I'm happy to announce our last speaker of today, Dr. Sylvain Tourel. He is working the project Gene Therapy at the Lead Discovery Center, and he will today answer the question, what is next up in life science for him? So, Dr. Tourel, the stage is all yours.
Oh, thank you. So 1 minute, I have to share the screen. This one. Okay, can everybody see the presentation?
Yes, it looks great.
Okay, so thank you, Bartels, first of all, to invited me to present some of my work at the Lead Discovery Center. I'm a little bit of an exotic person here in the mixture, because I'm not working specifically on microfluidic, but more on drug discovery. And we have been using, or I have been using some microfluidic self-design chip in order to push the drug discovery. So I will make a small introduction about the LDC. Nope, sorry. Yep. So LDC is basically a company which has been created from the Max Planck Society in the, I think it was 2008. And it's basically to make the link between academic research and advancing a project so that it will be suitable for pharmaceutical companies. So the problem of most of the academic research is that the standard of the experiment are not at a level where the pharmaceutical industry will be interested of. So that this is basically what LDC is doing to bridge or to close the gap in making projects more interesting for pharmaceutical industry. So LDC has over 100 academic partners. We have over 100 drug discovery projects. We have found already 19 leads which have been fully validated for specific diseases. Some projects are in collaboration with pharmaceutical companies. Some are basically internal projects. There is a lot of industrial investment which has been done in the last 12 years. We are reaching almost the 100 collaborate in the companies. So I will present you something today which we have that we are starting to use in the company, which is not a classical drug discovery component, but we think that it has great potentials. And even if in a few years LDC introduce antibody design and development, now I have introduced something called aptamers, which in our case are DNA aptamers. Just as a small information, aptamers are short-length molecules, either peptides or DNA sequences or RNA sequences. And we are talking about single-strand DNAs because they are capable of forming, like here, secondary structures. And this structure can specifically recognize target like protein or analytes and so on. Just for the record, for the people who are not biologists, the ribosome, which is the biggest machinery in the cells responsible to convert RNA into protein sequence, responsible for making peptide bonds, are made of RNA. And it is pretty clear that RNA is basically the ancestor of proteins. and they can make very complex secondary structure. Why we use DNA aptamers is because DNA is much more stable than RNA. And RNA degraded very easily because for example, our finger contains RNAs which degrade it. So basically, we use why it is for us interesting to use DNA aptamers is because, for example, you can use them for to make drug delivery, either with nanoparticle, like I do for my gene therapy project, but also to make like antibody drug conjugate or aptamer drug conjugate. This is basically an aptamer which will recognize one specific protein in the cell, and then you have a small drug in case, for example, of cancer. This is a cytotoxic drug which will kill the cancer cells. But we can use these as therapeutic tools or in diagnostic or as a laboratory tools. And this is basically now the area that I'm going to present you. Advantage of aptamers are basically that they are fully synthetic. They are much smaller than antibody. We are talking about 10 kilodalton compared to an antibody, which is about 200 kilodalton, so that they are much more flexible and much easier to fit in region where an antibody will never be able to log in. They are easy to storage because you don't need to cool them. They don't degrade easily, and they can be, you can apply it on a very large number of applications. One of the, I think here I have to empathize, the easy storage is especially regarding microfluidic and point of care diagnostic, is that in contrast to antibody, is that you don't have to cool them. So the transport from the facility, the production facility to the customer doesn't have to happen in a frozen truck or at 4 degrees. And in contrast to antibodies, the quality of one batch to the others is always the same because they have been made synthetically. And that's why we have been thinking in the past, why not working more on aptamers parallel to antibody to expand the range of tools that will help us to find new drugs. So for the people who basically don't know how to raise these aptamers, here is a slide basically presenting you the process which is called SELEX. It's a systematic evolution of ligands by expositional enrichment. So basically you start with a library containing a random sequence in the middle, and here you have at both ends sequence which remain always the same. Then you mix this library together with an analyte. It could be proteins, it could be small molecules. You incubate them together. The aptamers which bind to the protein will attach to the protein. The other one will remain in solution. Then you washing basically your protein, getting rid of the non-binding aptamers. Then you release from the protein the bounded aptamers, and then you re-amplify it using PCR. So then in the second run, you basically have a mixtures of aptamers which were basically binding to the substrate. Then you re-amplify it to using PCR to have millions of this single sequence, and then you again incubate it with the protein, wash the protein, elute the aptamers from the protein, and then so on. This way is basically you use the property that molecules binding stronger will basically release protein which doesn't bind very specifically. We are not using the classical SELEX because the classical SELEX was developed in the 80s. And it was basically very work intensive and resources intensive. You require 10 to 20 cycles. You required milligrams quantity of substrate, which is for some proteins not really a problem, but for some analytes or sometimes some protein very problematic, because you need so much cycle. It is very time required. It needs about weeks, and because of this, it is time and cost intensive. So the most expensive cost is basically the labor cost. From the binding, it doesn't make any difference if you're using the classical SELEX or the microselex. The microselex is a method that has been published, but that I have been improving in our laboratory. I'm not the exclusive one using it, but it works pretty fine in our hands. I need 3 to 5 cycles. I required only nanograms of substrate in order to have highly specific high binding aptamers. The selection happened within 1 week without the sequencing. And because of this, it is very time and cost effective. So the labor cost is the driving cost, but it is a factor a factor 5 to 10 lower than in a classical SELEX, and you still get KD in a nanomolar range. So how do we do that? So, and this is how a microfluidic can basically help us to overcome the main problem. When you do a micro SELEX, you want to basically improve the natural selection, binding selection of the aptamers to the substrate. It means that if you do a classical SELEX and you have you take a football stadium, and in this football stadium you have 100,000 aptamers, and in the middle you have 20,000 players, and you're trying to fit one aptamers to the players, to the 20,000 players. It means that you will find at least 20,000 aptamers, which are in high binding, middle binding, and low binding. So you select them, you elute them, you wash them, and then you go again and again. What we are doing is basically, instead of having 20,000 players, we have about, in comparison, 1 to 2 players. And then we do speed dating with these 100,000 aptamers, and we wait that basically if one aptamer binds on the player, on the substrate, and the binding is not very strong, and one is coming closer and he has a stronger binding, then you will kick off the one with a lower binding, so that at the end of the incubation time, and we are not talking about days, we're talking about 1 hour, then you only get aptamers which are in a high binding range. So in principle, the microfluidic works this way. You have here two channel connected to a chamber, and in the middle you have a small area that we coat, so that we basically can load the protein of interest on this very tiny dot. And then we wash in order to get rid of all the possible protein which were in solution. Then we introduce the aptamer mixtures. We incubate them for 1 hour. Then we use these 2 channel in order to wash basically the chamber. And then when we are done, we heat this small chamber at 95 degrees to unfold the aptamers. And at that point, the aptamer release from the protein and they are back in solution, but only the aptamers which basically were bound to the protein. And then we close this channel and push the solution in this chamber through a PCR meander channel, where you basically have 3 different temperature, and you basically do 15 cycle of PCR with primers, which basically are binding to the sequence of the aptamer, will remain always the same all the time. And then at the end of this, then you have a aptamer mixture, which is representative of the aptamers which basically were bound to the protein. Then we take this amplified PCR, we wash the residual primers, we refold the protein, we take a new chip, we introduce again a fresh substrate, and then we had the aptamer mixtures from the first cycle. And then again, we incubate and we run it like 3 to 5 times, so 3 to 5 chips. I can't show you the chips because this is an internal property design, and we are not intending to sell it and so on, so that we are not allowed basically to show any further design. But this is the main principle. The advantage of using the microfluidic is that basically we are working very low volume. We have no evaporation, so that we are not losing any liquids volumes. We have no contamination of whatsoever from maybe, you know, a technical assistant adding some DNA whatsoever. And we can basically being very reproducible in the process. It is very effective, and it's a very easy way to wash. Basically, we are not even using microfluidic pumps, sorry, Bartels, but we are using pipettes where we can basically fill volume or pipette out and volumes. But we are not excluding in the future to use pump for this purpose. We are using 1 chip per PCR, so we know that we are sure that we are not incorporating any contaminant, and the easy low volume ending make it very feasible to have this micro-select selection. Then we have to characterize them. For this, we are using something which is called an interferometer. You basically, when we do the PCR at the very end, we incorporate a biotin on the aptamers. We bind the aptamers on the streptavidin coated sensor, and then we have the protein of interest. And then we look basically if a signal is increasing. Just for the record, the interferometer is basically a biolayer sensor. If a biological layer is increasing at the end of the sensor, then the frequency from the light coming back to the sensor change, and you can say, you know, how big the layer has been increased or decreased. This way we can run binding curves, and we can determine binding constant, which are for us very which is the most information, because we usually look for aptamers which are competitive or comparable to antibodies. And basically, sometimes we try to find aptamers which block the interaction of 2 proteins, so that basically we attach first the protein on the sensor, then we put the aptamers, and then we look, does the partner proteins bind to the sensor, or does the aptamer block the binding on it? So this way we have been able to raise 30 different aptamers, urban cancer markers, and now I will basically show you one application, how we use these aptamers against specific cancer markers. So the main problematic in fluorescence microscopy, which is the way to go when you screen for cancer markers in biology, is that you basically use antibody recognizing specific cancer markers, and each antibody contain a different fluorophore. Here is basically you see below, this is the different fluorophore that we can use. And depending on what antibody with what fluorophore is binding to what protein, and you get different pictures, and you can evaluate, okay, what is the stadium of a cancer, or the protein is located in the nucleus or on the surface or whatsoever. The problem is here. You have only a certain level of fluorophore that we can use, and each fluorophore required a set of filters in order to discriminate what fluorophore we're actually looking at to get such nice pictures. And it means every color means one set of fluorophore. And most of the fluorescence microscope that we will use, you can maybe have 7 filters. More than 7 filters, it gets very complicated. It is complicated to find the protein with the right fluorophore, and it's so people start to label the protein, the cells separately, and try to to understand, okay, where does the protein is being expressed, and how does it correlate it from one cancer marker to the others. So, like I said, there's a limitation in terms of availability of those fluorescence filters, the limitation of the antibody available on the market, and most of the time, 7 different antibody is already a big challenge. So, and now we have came up with an alternative process that we are working on based on aptamers. So let's consider you have made 5 different aptamers recognizing 5 different proteins on the cell surface, so 5 different cancer markers. The green and the blue sequence are basically always the same. Only the middle region is as a different sequence, and this sequence is based on the SELEX process we have been made than before. So we incubate the cancer cells together with the aptamer mixtures. I will not call it a library, because a library will be a lot of random sequence. Here, all these sequences basically known. So it's aptamer mixtures. We incubate them. If the cancer marker is that the aptamer recognizes, the aptamers will bind to it. In this case, this is the brown one and the pink one. The other one will basically remain in solution. We wash, like you will do for antibody, to get rid of the non-binding aptamers. Then, like on the selection process, we hit the sample in order to release the aptamer and having them free in solution. And then we do a PCR. So at the end, we amplify the sequence which were bound to the cells. The problem is that you end up with a mixture of aptamers, only the one we were binding on the cells, but then you need a way to discriminate which aptamer you have done in solution. They have exactly the same size, and you could basically make a next generation sequencing, but it will be more time consuming and much more expensive. And what we do is basically something which is called hybridization. So the vertical line here with the dash is basically the complementary sequence of this red sequence. So it means that we use the fact that when you have 2 pieces of DNA which are complemented to each other, that they are capable of forming a duplet, like double helix. for our genomic DNA. And basically, we have an array of 5 different dots, in this case, with the complementary sequence of the aptamers sequence, which are known to recognize the cancer markers. And we incubate this mixture. And here you can see we got only brown and pink, and then they are binding to the pink and to the brown. And basically we see 2 spots glowing, telling us that this and this cancer marker has been found. But at the same time, we can also do quantitative analysis, like it has been mentioned in the last slide. It basically, depending on how many cycles you are doing, the more concentrate the substrate, the faster you get an amplification. And it means after, let's say, 15 cycles, if the aptamer is present, then and it has been amplified, then you will have a high quantity of aptamer in your sample. If the concentration was much lower, then the exponential amplification will happen a bit later on, so you will have less sample, and this way the intensity of the dot will tell you how much cancer marker you had on the cell surface, because it is directly correlated to how much aptamer do you have. We do it also, the hybridization in a microfluidic chip, because it speeds up the process. Again, we are not losing any volumes. When we are working, usually when we incubate with cells, we are working about with 20 to 100 cells, because cells are very expensive, and if you use much more cells, then you have to grow them, and some cell line doesn't grow very easily. And because in the long term, we are thinking of using patient cells. So if you take, for example, a patient has a cancer and you take a biopsy, then getting 20 to 100 cells of a patient, it is not problematic. If you need 100,000 or 1 million of cells, you are dealing with much bigger problems. Okay, so that the advantages of putting 20 to 100 cells on the microfluidic is that the flow is not so strong, so the cells can attach very easily. You can wash them very easily. This is very easy to handle. So we do the binding in this chamber, like we did the SELEX process on the true substrate. And then when we're done, we incubate with the aptamers, we wash the aptamers, and then we incorporate the PCR mixture. We run the PCR, and then when we are done, we push the liquid from this chamber through this chamber very slowly, and basically you give the mixture time to interfere with the complementary sequence and to have an hybridization. And then we look what dot is basically showing either color or a fluorescence signal. And this gives you, for example, in this case, we have 6 dots in the horizontal times 4. So it means we will look to 24 different cancer markers, and we can discriminate all these 24 cancer markers within 1.5 hours. Here is, for example, a primary data that we are working on. We were using nitrocellulose where the complementary sequence was spotted, because this is more easy to attach on the nitrocellulose membrane than on the chip, but we are moving forward to moving on the chip. Here you basically have the positive control is to make sure that every single aptamers that we have been tested are do the hybridization. And here you have different cell line where we have tested these 8 different aptamers. And you can see, for example, in the cell line number F, we found the cancer markers number 5. Here in I, number 1 and 2, or G, 1, 2, and 7. So this way, by getting, we are trying to get the fingerprint of the cancer markers of the different cell lines. I have to say that currently in the diagnostic for cancer at the hospital, is most of the time, if a tumor has been found in the breast or in the liver, then the pathologist will test only a certain number of markers which are correlated by tradition with this type of cancer. So it means if you have lung cancer, they will use, for example, EGFR, or breast, that will be CD44, and so on. The problem is, by doing that, is that you if you're lucky, you will find these markers, and then you're sure, okay, I'm dealing with the breast cancer or with the lung cancer whatsoever. But if you don't see anything, then you don't know. You might have other cancer markers. The biopsy of the patient doesn't last forever. Even if you freeze it, you don't know by when you thaw it if the sample will remain viable. So having technology where you can basically screen systematically a high number of cancer marker, even if these markers is non-correlated to lung cancer, but you found the tumor in lung, we'll have the advantage that we will more understand about the cancer biology and being able to improve the point of care diagnostic in cancer, and correlate, for example, what pattern, so what fingerprint was successful with what drug. And since we understand what cancer markers, what is the biology of the cancer markers, and what cascade are activated in the cells, then we can even think about, well, this marker is active, then maybe we can block this protein, and this way we can kill the cancer cells. So here it is really important to fully recognize the pattern of a cell surface, even because the cell surface make the interaction with the other cells, or with the interface of cells and serum and blood and so on. This work is a very early work, but it looks very promising, and I think we will be able in the coming 2 years be able to raise over 50 different aptamers, and our goal is basically to develop a methodology where the methodology we have it, but to have an aptamer mixtures where we will be able to discriminate 50 different cancer markers on patient samples. These methods, they can be applied on bacteria to characterize what kind of bacteria. We are working on a project to discriminate the type of cancer, or the phenotype, the type of viruses, the phenotypes of viruses. You can detect bacteria and a lot of different molecular substrate or biological substrate. Something that I found very interesting to the presentation, which has been shown before on this nanodrop, where 2 antibody was used in order to recognize, for example, protein-protein interaction. These antibody were labeled basically with DNA as let's say as a barcode. Here, you don't require this because you could use directly 2 different aptamers labeled with 2 different fluorophore, and then you get the same analysis. So maybe that could be an improvement to the methodology which has been shown in the previous presentation. Here, so I'm getting at the end of the presentation. Basically, at LDC, we are looking at the overall biology of disease, and we understand very little. Yeah, and the more we have tools to analyze biology, the more questions are popping up, and the complexity of biological system required different methods to basically get different answers, and aptamers are surely one tools which will get more focus in the future. Understanding the cell on the molecular level, like I say, will be for us more easier to define one target is the most suitable for a new drug. Since aptamers are behaving like antibodies, they are really multifunctional and very flexible, and they basically speed up our drug delivery process, and is a real add-on. And here, to make the link to the conference, because it's all about microfluidic, and we have to say that microfluidic chip has helped us basically to overcome the problem of handling this little volume. So here we don't have very complicated chip, and we don't require a lot of pumps and valves and so on, but it's showing basically that how powerful microfluidic can be for some specific application and really help in any sense. Yeah, so thank you very much to listen to my talk. Here's the group of LDC, and this is basically all the partners that we had since LDC was created. Thank you very much.
Thank you, Dr. Tourel, as well. Thank you. That was very informative for us all. Since we're over time, I just want to throw in one quick question for you, and regarding the aptamers or microselex method. When do you think something like that will reach the market or a broader scale of use?
So I have to say, so SELEX was developed in the 80s, in 83, from a professor in the Harvard Medical School. And he has not came to the market because the PCR technology was basically at that time something which was used only by a very little number of people, because you required a lot of enzyme. The synthesis of oligonucleotide was expensive, complicated, and so on. Then the antibody market in that time raised a level that nobody was thinking of using aptamers. And most of the people were focusing on RNA aptamers, which are more complicated to deal with. But since the human genome sequencing project was going on, all the PCR reagents, the synthesis of oligos, the sequencing analysis, and all these techniques have improved and have become much cheaper. And since in the last 10 years, basically people came back and looked at aptamers and say, oh, actually it was interesting. Why we have not been using it? And right now we really.
Ich suche nach basically PayPal Arkenberg in Luca hat ein
Sorry,
Athenes.
Siri is big watching. So people are watching back and take aptamers as a novel alternative. So right now, if you look at publication, they are making up to 1 to 2% of publication regarding new, new cancer markers detection, but they show up more and more in diagnostic tools. So I think in the next 5 years there will be already a lot of aptamers as an alternative to antibodies. And again, the main advantage in diagnostic is that the aptamers doesn't have to be cooled, so that you are sure when it's arriving at the point of care where it's going to be measured, that the aptamer will be fine for the measurement. You know.
Okay, thank you very much.